So in Chapter 6, we're going to discuss microbial growth. It's going to be something that I've already introduced to you. We'll talk about how they actually grow. We'll talk about the process of binary fission. We'll go into a little bit more detail on what we talked about. I've already introduced you to the growth curve in Chapter 8, and once we go through that again, I will show you the different physical and chemical requirements for growth. And lastly, we'll go over the different types of media. So if you look over here on the right, this is just an example of chemically defined media. All this stuff that's in this media, the glucose, ammonium phosphate, sodium chloride, magnesium sulfate, potassium phosphate, and water, they're there for a specific reason. And as I go through the different physical and chemical requirements for these different bugs, you'll understand at the end of this lecture why each of those things is actually in this chemically defined media and why E. coli is able to grow in that. And so these microbes won't be able to grow unless you meet their minimal growth conditions. And each microbe is going to have different minimal growth conditions. So it's actually really, really hard to get these microbes to grow. In fact, we can grow less than 10% of the microbes out there. So when you look at this picture on the bottom here, where you see this person has a wound on their foot, you got to figure out, if you're in the healthcare field, you're going to be part of the team that helps figure out what the heck the problem is with this person, okay, why they're experiencing this infection, and more importantly, how to get rid of that particular infection. In order to do that, you have to be able to get these bugs out of here and get them onto here. Okay, and as I said before, we can grow less than 10% of the microbes out there, so this is actually a really, really hard thing to do. Most of the time, we don't know what it takes to grow these microbes. So hopefully, this microbe causing this infection right here in this wound is one of the less than 10% that we can actually grow, so we'll be able to figure out first what it is, and eventually, hopefully, how to kill it, which we'll talk about a little bit later in this unit. Now, with these microbes, we measure growth a little bit differently than in these microbes than we do in us. So a lot of times when you have little kids, what will happen is you put them against a wall and you draw a line every so often, every year, and you say, okay, Jacob was this height this year and this height this year, and he grew 2 inches over the last year and stuff like that. With these microbes, it's a little bit different because when they undergo binary fission, they are going to actually double in size, but they split down the middle, so in the end, they're the same exact size that they were to begin with. So we don't measure microbial growth in terms of how big these cells are. We actually just look at the number of cells. So if we look at these two plates here, the reason why we know microbes are growing on there is because, well, there's several reasons. One of them is very apparent. You can see from the tubes on the left, they are cloudy. They originally probably started off clear because most of the different types of broth are clear, and as the microbes grew in there, they got very, very turbid, very, very cloudy. And you can see the one on the left actually changed color, so it's nice and red. And the one on the right is a little bit yellow in color. Over here on the plate, you can see all these different colonies that are out here. Okay, and you can see a lawn over here. What do we call a lawn? It's just continuous growth of all these microbes. There's a lawn over here too, and in this section. And you can start to see it starting to break up over here. We can see some colonies still attached to one another, and then we finally get these ones that are all by themselves out here. Okay, we'll be doing this. Actually, if you're in my hybrid, we'll be doing this a little bit later in the semester. If you're in my traditional class, you've already done this streak plating that's there. But with these colonies here, they're just a bunch of cells that have undergone binary fission, and they're just piled up on top of one another. Each one of these colonies, if they're separate from the others, started from a single cell. And then underwent several rounds of binary fission and they increase the numbers. Other ways you can tell if microbes have grown, we'll see a little bit later, gas production. So imagine you're sitting there on the side of the road next to a dead animal. All those things that happen to a dead animal, a lot of them are actually indicators of microbial growth. So you look at the smell. A lot of times when microbes are growing, they don't smell very good. They'll be breaking down their source of nutrients, which is going to be that dead animal, and producing these molecules that smell. It's the same thing underneath your armpits. Okay, they're breaking down some of the molecules that you release in your body, and they produce these chemicals that smell really, really bad. Other things like gas production. If you look at a dead animal, a lot of times what happens is if it's left on the side of the road, it will start to expand. Some of them actually even pop, and the insides will come out whenever they pop because of gas production. If you look in the news every so often, you'll see stories of beached whales and huge animals from the sea that have been beached. And there was actually a story which I thought was pretty funny. I think it was somewhere in Finland or Switzerland or something like that. There was a beached whale over there, and It was starting to decay, it was starting to smell bad, it was starting to affect their business on the beach, and so what they decided to do was they decided to go dispose of it in the city dump. So they loaded it up, they got a big crane, loaded it up on the back of a 18-wheeler, and in order to get to the dump, they actually had to go to the center of town. So this whale had been sitting there for a while, had been decaying, and the microbes inside of it had been growing and producing a lot of gas. So right in the middle of town, what happened is the 18-wheeler hit a bump, and there was an explosion. All that gas built up inside the whale, and the whale just gave way. The skin and stuff just gave way, and so the whale exploded in the middle of town. And you can just imagine the smell of dead whale all over the place, dead whale insides all over the place in that town. So they had a heck of a time cleaning that stuff up. But sometimes animals do explode whenever they are filled with gas from the microbes, because as microbes metabolize, what they do is they fill up the insides of an organism with gas. Dead people do the same thing. Whenever they die, their normal flora doesn't die right away. Right? It's got a nice source of nutrients there inside of this person, so it's no longer being kept in check by the immune system, so it just starts to grow and they'll start to build up gas as well. And so dead people actually will fart because of all the gas that's inside them. It's kind of weird, but it does happen. Now here's binary fission in a little bit more detail. So the first step in binary fission is the replication of the genome. And you can see in this cell there are now 2 copies of the single chromosome. You see in this section right here, it's actually that chromosome is actually attached to the side of the cell. And so after the replication of the genome occurs, what happens is everything else inside of the cell replicates. And since these chromosomes are attached to the side, as the cell doubles in size, what happens is these will, because they're attached to the side, they will move to one part of the cell or the other part of the cell. They'll just be separated from one another. So this is actually a lot easier than what happens in eukaryotes, because if you remember in eukaryotes, what happens is you have prophase, metaphase, anaphase, telophase, and you have all these spindle fibers and stuff involved in the division of the nucleus. But over here, basically, it's just the chromosomes attached to the side, the cell gets bigger, it elongates, and that is the motion that actually separates the 2 chromosomes. Then there's a ring of proteins that's going to form right in the middle here, and it's going to pinch the middle, okay, and that's going to divide the 2 cells. And this last step over here, the cells separate, that's actually optional. So if you have 2 rods, there sometimes they will separate to one another. In other cases, they won't. They will stay stuck together, and the arrangement would be diplobacillus. Now, if you look at this process of binary fission, it's actually an asexual process. And so you go from one cell, copy of the genome, to 2 cells that are genetically identical to one another. And then these 2 cells will keep on dividing, and so you go 2 to 4, and it's just exponential growth from there. So they can actually grow very, very quickly through binary fission. Just a very simple process. Alright, so the amount of time it takes for the parent cell to go through binary fission and go into 2 cells is its generation time, and it varies depending upon a lot of different things. Okay, some microbes are very fast growers, some microbes are very slow growers. If we look at this table right here, we've got the microbe name in the first column, temperature, their optimal temperature of growth in the second column, in degrees Celsius, and their generation time at that particular optimal temperature, and on the right column. Now, as you look down, you'll notice there's 37 multiple times here, and I don't want you to go away from here thinking that every microbe loves 37 degrees Celsius. The reason why 37 is mentioned so often in this table is because this is medical microbiology, and we're dealing with a lot of bugs that can cause disease in us or grow on inside of us. And so that's why they're there. Some microbes don't like it at 37 because it's too cold. Like, for example, Bacillus stereotomophilus. Optimal temperature is 60 degrees Celsius. That's really, really hot. And at that particular temperature, it's a really fast grower. It divides once every 11 minutes. This bug causes food spoilage. And so it'll love to grow on your food when it's still hot. A lot of times think about what happens with your food. You are at a barbecue, the food comes off the grill, you put it in a plate or a pan or something like that, and you set it on the table, and you're thinking, well, it's too hot. Microbes won't like it on there. But that's not true. And some of the microbes will love it at that elevated temperature, and they'll be growing on there, dividing like crazy. As it cools down below 60, all that's going to happen is Bacillus stereotomophilus is going to grow a little bit slower than the 11 minutes, sometimes a lot slower, depending upon how cool it is. E. coli loves it at 37 degrees, and at that particular temperature, divide once every 20 minutes. Relatively fast grower. With E. coli, it's almost exclusively found in the intestines of mammals. It's an indicator of fecal contamination or sewage contamination. And if you think about where that contamination is, it's pretty much all over the place. So any place you sit your butt down, fecal contamination is going to be there. And if you think about your clothes, a lot of people think, well, my clothes prevent fecal contamination from occurring. But if you think about it, look at even your jeans and all your clothes. If you hold them up to the light, you can actually see the light through those clothing items. And so there's huge holes in there. I say huge holes. We can't see through them usually, but the microbes, they'll be able to fall right down through those huge holes because remember, they are very, very tiny organisms. So even if you have multiple layers of clothes on, they'll be able to get through, and so your chair is covered with fecal contamination. And then, so basically, anywhere you sit your butt, you're going to have fecal contamination. Also, where you put your hands. Because crap gets on your hands. A lot of people don't like to think about it, but it does happen, right? And sometimes it happens where you get a lot of crap on your hands. So, for example, when you experience toilet paper failure, it happens, right? The toilet paper fails to protect your hand from the crap as you wipe your butt. And so you get a lot of crap on your hands. A lot of people think, well, I'm just going to wash my hands, I'm going to get rid of all that crap. It's not true. Hey, if you think that's true, next time you get crap on your hands, I challenge you to wash your hands and then smell that area of your hands afterwards. And what you're going to smell, pretty much guaranteed, is going to be soap plus crap. If you use soap, hopefully you did use soap, and you're washing your hands correctly. But you're still going to smell that crap. You can wash it usually a couple of times, and if your nose is sensitive enough, some of y'all have dead noses, but or a dead sense of smell. But if your nose is sensitive enough, you can still smell that crap after a couple of hand washes. And so that E. coli is going to be there because it's in high concentrations of feces. It's one of the most commonly found microbes in feces. And it's usually the second member of your normal flora. The first member of your normal flora is usually lactobacillus acidophilus. So in response to the hormones that are released when a mom is about to give birth, lactobacillus actually goes crazy. It starts growing like crazy, going through binary fission. And as it goes through binary fission, it's preparing the baby. It knows the baby's going to come out. And so as it comes out of the vaginal canal, lactobacillus gets on it and does three things: takes up space, uses up nutrients, secretes helpful substances. As I said, E. coli is usually the second member of normal flora. It's going to get on the baby whenever people start touching it. Pretty much as soon as it comes out, people start touching it. It's like, let me hold your baby, all this kind of stuff. Let me kiss the baby and pinch his cheeks and all that kind of stuff. And so, I mean, it's a necessary thing to get these microbes on the baby. And so getting that E. coli on inside the baby is very, very important. One thing you should not do is you should not let anybody that's sick touch your baby whenever it first comes out. Okay, a lot of people are like, well, it's good for their immune system. They need to get exposure. It's like, but they don't need to get exposure right away. You let them have a little bit of time to actually build up some of their defenses before they get exposed to some respiratory tract infection. Okay, because respiratory tract infections can be very, very dangerous to babies when they first come out. They don't have very much strength. Right? They have this huge shock. They were inside a womb and then they come out. And so they are very, very immunocompromised. So keep those sick people away from your babies. You don't even let them in the room. I know one of my cousins was sick. Her daughter was sick whenever Samuel was born, and she didn't come in the room. I didn't let her come in the room. And she was mad, and the little girl was, I think she's like was two at the time. She wanted to see Samuel, and she was crying out in the hallway, but I was like, no, you're not going to come in. You're not going to get him sick. He is too valuable to me at this particular point in time. And her mom got all mad, but I was like, I really don't care if you get mad. This is my son's life that we're talking about, so you're going to do what I say. So you just have to take that kind of attitude with them and protect your babies as much as possible, because there are some horror stories that I've heard about babies dying from people that actually come in with respiratory tract infections. So make sure you protect them. It's your job to protect them. This next bug down here, Mycobacterium tuberculosis. The current outbreak for TB, actually, scientists estimate it started where they first recognized it somewhere around 1933, I think it was, or so. Okay, and it's still happening right now, and it's actually getting worse. In many cases, what actually happens is when there's an outbreak of disease, that people get sick, and then one or two things either happen. They get better because their immune system kicked in, and a lot of times they develop immunity, or they die, right? And so you're losing, that pathogen is losing the number of hosts that it can actually multiply in. But the whole thing about Mycobacterium tuberculosis is it grows at a very, very slow rate. You can see it divides once every 6 hours, and the infection can last for a long time. So for some people, when they're treated, they'll actually have it for the rest of their life. and they will be, during certain stages, they will be contagious. So it's not like the flu. A lot of times we're about to enter into the flu season, and so in the beginning, what happens is this flu spreads like crazy because nobody's had it, or there aren't a lot of people to get the flu shot. But then after a while, a lot of people have had it, so it doesn't have very many hosts to go into because there are a lot of people that are immune around it, and so it doesn't spread. And the actual outbreak or epidemic actually slows down and eventually stops. But with Mycobacterium, that's not the case because it's just such a slow grower. And we have so many different strains out there. I think it was like a little bit over 10 years ago, there was a new strain of TB came out. It might have been 15 years now or so. But it's called MDR-TB, multi-drug resistant tuberculosis. That one popped up out of India. Okay, we'll talk about in a little bit why these strains seem to pop up out of India, because it really, really sucks. But so this multi-drug resistant means it's resistant to more than one drug that's commonly used to treat tuberculosis. The second version that popped up out of India was XDR-TB, extreme drug resistant tuberculosis, and that was resistant to multiple, or a lot more than MDR-TB. And nowadays, the new one that's come out in the last couple of years from India is TDR-TB, which is total drug resistant tuberculosis. Okay, with that one, we can't treat the people that actually have it. We don't have any drugs for that. Okay, so one of the reasons why this bug is hard to treat, the treatment time for tuberculosis is usually 6 months or longer. If you've been exposed to this bug, a lot of times it will give you at least 3 months worth of antibiotics. Okay, but if you actually have it, it's going to be a long antibiotic regimen to actually deal with it. And it's going to take this very, very seriously. Some cities actually have people that will come out, social workers that will come out and make sure that you take your drugs. They will bring your drugs to you wherever you are, watch you take them, because there are so many different versions out there, and a lot of the versions are popping up because people stop taking their drugs because it's such a long treatment. But one of the reasons why it's hard to treat is because it's such a slow grower. But it also has some other characteristics we'll talk about a little bit later. This last one down here, Treponema pallidum, causes syphilis, we talked about. And you can see it's a super slow grower. It takes about 2,000 minutes for it to grow. It causes syphilis, which is an STI. There are about 40,000 new cases of syphilis that are reported each year in the United States. And that disease actually has a history, a long history. A lot of times in the past, sailors, okay, we're talking about like Columbus time, stuff like that, the sailors actually considered syphilis a badge of honor. And we're like, you aren't a real man until you actually get syphilis because you haven't slept with enough women, kind of thing. Which is a really, really stupid thing, but that's just the way that they thought back then. Alright, so here's the growth curve as we talked about it before. You can see that there are 4 different stages that cells go through over time. On the y-axis we have the number of cells, and the x-axis we have time. So we're just going to discuss it as these cells would go through this growth curve if we inoculated a tube. So we're starting with stage 1 right here. And what happens at this particular point in time are we inoculate our tube with, let's just say, usually we inoculate with a single colony. They enter into lag phase. The first thing they're doing is they're trying to decide whether or not conditions are good for them to grow. So are their minimal growth conditions met? It's either yes or no. If the answer is yes, they'll go through the rest of these steps. If it's no, then they won't. They'll stop right there and they won't do anything else. Okay, so if it's yes, the second thing they're going to do in lag phase is they're going to prepare to divide. So they replicate their DNA, replicate everything else, and so you see this is a flat line. They're actually not increasing in numbers yet. Conditions over here are going to be good. And they got tons of nutrients, very little waste. And so in our little test tube, so of course they're going to decide yes if we're capable of growing them, and then they go through after they get it in from or end up at the end of lag phase, they enter into our second phase, which is log phase. And you see exponential growth here. There's a lot of metabolism going on. They're dividing as fast as they can. They've got tons of nutrients, very little waste. And at this phase right here, during this phase right here, since they are highly metabolically active, they're actually very, very sensitive to our treatments. So it'd be nice if when we treated an area, if they were super metabolically active, because our treatments target metabolism in a lot of cases. And you see their numbers are increasing very fast. And because of that, they're using up the nutrients, producing a lot of waste. So over here in this area, there's going to be very little nutrients and lots of waste. So the rate of growth slows down a lot, and they enter into stationary phase. The amount of life equals the amount of death, and so they're trying to survive. And in chapter 8, we talked about how some of them will undergo transformation, taking up naked DNA so that hopefully they can become different enough to try and survive this new environment. But as they continue to grow, what's happening is most of the nutrients are gone, and there's going to be a lot of waste. So it's really unhealthy, and they'll enter the last phase, which is death phase. They don't have any nutrients. There's a ton of waste. And as you can see, that there's a rapid rate of growth, or a rapid rate of death. Sorry. So a lot of them die very quick, and then that rate slows down. It's very important to realize that the weak die first, the strong remain behind. Okay, especially when you're talking about treating an area, and you're treated effectively, when trying to kill all those bugs, you don't want to take that treatment off before it has time to work. You want to give it that effective time. Same thing with antibiotics. You don't want to stop taking your antibiotics until you've completely finished with them. It's very, very bad because you'll be encouraging resistance inside of you. All right, so when it comes to the different requirements for growth, we're first going to talk about physical requirements. The first physical requirement is temperature. And based upon what temperature they like, we've got them grouped into, we're going to say 3 different categories. Even though we've got these 2 microbes here, we're just going to lump them together, okay? So if you look at this little thermostat right here, you can see in the middle here is what we call the danger zone. So that's like in areas that are very, very close to our body temperature, or areas in this temperature range that we can stand, or where we're normally found in, like room temperature is going to be somewhere in the 20s. Our body temperature is going to be in the 30s. Different areas of our body are going to be in different temperatures. All right, so our first group is the thermophiles. They like it hot. They usually like it above 45 degrees Celsius. So they're going to be way up here in this region. Okay. Our mesophiles are going to be right in the danger zone. Okay, we'll have a lot of mesophiles that are pathogens. And then our psychrotrophs, psychrophiles, we're just going to say they usually like it below 30 degrees. They're cold. The psychro means cold. So they are cold-loving organisms. Okay. And you'll see that a lot of times these guys are going to cause problems in those foods that we tend to refrigerate. As I said, our thermophiles like it hot. They'll like it in temperatures above 45. Some of them like it at 100 degrees Celsius, where water's right around boiling. Some like it at 120 down at the bottom of the ocean. There's so much pressure, the water does not boil at that particular temperature because of all the pressure that's there. So in all these different environments that are really hot, you will find these thermophiles that are there. Their proteins are not going to be denatured because they've evolved to work at those particular temperatures. So through years and years of evolution, their proteins probably have stronger bonds holding them in the appropriate shape. They don't rely so much on hydrogen bonds or van der Waals interactions, things like that. They'll rely more on covalent bonds, ionic bonds, to keep those proteins in the right shape, because their proteins are going to be made up of the same amino acids, so they need stronger interactions holding them in the appropriate shape. So if you think about why would a bug, or why would a microbe like it here? It's like, well, who else is going to be there? Nobody else is going to be there. If a microbe, like even if you actually go over there and you get in 100 degrees, you're going to die. And what you're going to do is you're going to become nutrients for all of these bugs that are there. There's tons of nutrients already in this area because nobody else is there using them. It's only going to be the thermophiles that are using them. So they found their own little niche, their own little place where nobody else bothers them, and they're able to grow just fine at that temperature. Then we have the mesophiles. The mesophiles like it, you can see between 25 to 45 degrees Celsius. Our body temperature falls in this range. You can see in this thermograph here, they were usually only at 37, right around the core of the body. And as you get into other areas, will significantly cool down in those other areas. So you'll have different bugs growing in different areas depending upon what temperature they like. Now, just because it says that they like to grow between 25 to 45 doesn't mean that they're going to be, all of the mesophiles are going to be able to grow in that temperature range. Some of them are very specific. Like, for example, E. coli. E. coli it loves 37 degrees Celsius. It can tolerate temperatures that are colder than that. But when you get too high above 37, what happens is E. coli's proteins denature and it dies. So if we were to take a culture of E. coli, grow it at 37, it would do nice. It would be growing once every 20 minutes, dividing once every 20 minutes, and it'd be nice and happy. You take that same culture tube, put it at 42 degrees Celsius, and 90% of those cells will be dead just like that. Okay, very, very quick because the proteins denature. and they're not carrying out the roles that they need to carry out. Okay, so some of them aren't able to grow in all these temperature ranges, but with the mesophiles, this is where you find the most pathogens because our body temperature, the different temperature ranges within our body fall within this category right here. Some mesophiles are actually special in that they can withstand high heat for a short amount of time. Okay, and those are called the thermodurics. So duric means strong, thermos has to do with high temperatures, and so they'll be able to withstand that high heat for a short amount of time, so they can withstand things like pasteurization, most forms of pasteurization, not all forms of pasteurization, but most. Now the only thing that's good about these thermodurics is that there are no known pathogens in this area or in this group yet. And so if we look at, it's not just our body we have to worry about, it's whatever's around us. And so most everything around us is going to fall into this temperature range, 25 to 45 degrees Celsius. So when you are working, you need to make sure that you're not touching all these different things around you. Minimize your actual touching of everything, because especially when you're trying to be clean and you have gloves on, don't put your hands on the countertop because you will be picking up bugs, and those bugs can potentially produce disease in your patient. So if you think about your actual work environment, you should, as I said before, you should be working like you are a germaphobe. So you know that phrase we've been talking about before about how less than 10% of the microbes out there are pathogenic? That number does not hold true in a hospital. It does not hold true in a doctor's office. Okay, why? Because there are tons and tons of sick people going to that place. And if you think about what sick people do, their nose is running. Okay, they don't always have Kleenex, so what are they going to do? They're going to use their hands, and they put their hands on the armrests in their chairs. Or they're going to be coughing, they're going to be sneezing. When they're sick, their body's trying to get rid of these germs. It does not care where those germs go. All it cares is that they're out of the body. So you're coughing and sneezing all over the place, and every time they cough and sneeze, they're going to put germs all over the place. Sweating can also spread disease. There's a bunch of different ways that pathogens can be spread. So these places that you're going to work, since you are going in the healthcare field, they are going to be filled with tons and tons of pathogens, especially during cold and flu season. Probably again, every surface of that doctor's office is probably covered with the flu virus during flu season, especially primary care physician office, because so many people go in when they have the flu. So um, it's one of those things where you don't want to go into a doctor's office during that season because you are very likely to get sick. And the last group we have are the psychrophiles, psychrotrophs. They like to grow between 0 and usually 30 degrees Celsius. A lot of these bugs, as I said before, they will be able to cause food spoilage. I think we put food in the refrigerator because they're low temperatures and they slow down the growth of these microbes that are on that meat or on whatever food you actually put in there. But some microbes will still be able to grow at those slow temperatures. So you think about, like for example, your cold cuts, your hams and turkeys and all that kind of stuff. They have a lifespan. Okay, they're not good forever. Or chicken. Okay, if you aren't using your chicken the next couple of days and you just leave it in your refrigerator, chances are it's going to go bad. You can't leave it in there for 5 days and expect it to stay good because there's too many microbes on there. There's too many psychrophiles that even though if you put it in the refrigerator, they are still going to be able to grow. So let's talk about some of these. Pseudomonas. We talked a little bit about Pseudomonas when we talked about assimilation plasmids. Burn victims have a big problem with Pseudomonas. So if they are burned over a huge area of their body, they are really immunocompromised. You're lacking the normal flora in that area, they're lacking their skin in that area, and a lot of these burn victims will succumb to Pseudomonas because it doesn't have any competition. Pseudomonas is able to grow in various areas of the body. It can cause wound infections, respiratory tract infections, septicemia, endocarditis, urinary tract infections. So basically, whatever gets inside the body, it's going to be able to produce disease. And so you got to try and make sure that it doesn't cause that infection in your patient. Make sure that you are not using quats as a disinfectant because certain strains of Pseudomonas will be able to break down the quats. Quats is Quats. And we'll talk about some quats in chapter 7. But certain strains of Pseudomonas will have assimilation plasmids that allow them to break down the quats and use them for nutrients. And so you think you're disinfecting, but you're actually just feeding this bug, and you're feeding the bug that's going to cause a problem for your patient. Another group of patients that has a problem with Pseudomonas is going to be those that are on breathing machines, ventilators. You think that tube is going down their throat, and there aren't any defenses in that tube. So Pseudomonas gets down there, it's going to grow inside of their lungs. And the best way, when Pseudomonas grows, it grows as a biofilm. So the best way to remove it is by scrubbing. Scrub it off, and you really can't do that in the lungs. They do it for burn victims. You imagine how unpleasant that is to have to scrub some of those wounds and actually carry out debridement, which is removal of the tissue that's infected. Even it's one of those things where if a person is already on a breathing machine, they're pretty bad off to begin with. So you get this infection, it causes pneumonia inside of their respiratory tract, and they die from it. A lot of them will die from that. So those patients that are really immunocompromised have a problem with Pseudomonas. With Listeria, there's been a bunch of outbreaks of Listeria. Okay, the last one was actually the bluebell one. I need to actually update this slide here. But before that, it was this counterlope thing where it caused 31 deaths. With the bluebell one, it's estimated that the actual infections go back all the way to 2010, the last time I checked. But it's one of those things where we're now learning about all the different horrible conditions that were present in the bluebell factory. Safety was not a major concern. Getting rid of sources of water or moisture was not a major concern. So they had certain areas where the air conditioning vents were leaking, and when they would leak, they would leak into the line, into the vats of ice cream and stuff. And that is a nice place for different microbes to grow. For some of these machines that they use to actually package the ice cream and put it in their containers, they knew that it was infected with Listeria, and they still continued to actually use those machines. Okay, and they didn't really stop until people started dying and people started suing them, which really, really sucks. Because there are a lot of people that are now coming forward and saying that this problem lasted for a long time. And what really, really sucks is when these people come forward and you look at what they've been through. Like some of these people have lost fingers to some of the machines in Bluebell. So with Listeria, it's a terrible situation because these people that died were usually already in the hospital. They were already immunocompromised, had some sort of underlying health issue, and then they get in the hospital, they eat some ice cream, and this bug gets inside of them, and they die. Okay, Listeria can cause encephalitis, meningitis, and cause all kinds of infections. I don't think we are actually ever going to know the final death toll from the Bluebell outbreak from Listeria, because you think about what else Listeria can cause. It can cause miscarriage, spontaneous abortion of babies. And how many pregnant women like ice cream? Gotta think, it really, really sucks. Okay, how many pregnant women miscarried because they ate Bluebell, they contracted Listeria, Listeria caused the miscarriage. So it's a pretty sad situation. Really, really sucks that they actually didn't pay attention to the warnings that were there. Now the Bluebell factories have to go through so many different checks before they can actually send the Bluebell ice cream has to go through so many checks before they send the stuff out that it's not going to have Listeria in it. Hopefully, if the FDA is doing all that it says it's doing. All right. The last bug here, Clostridium botulinum. Clostridium botulinum causes botulism. And botulism is one of those diseases where this toxin here, the botulism toxin, when it's ingested, when people get intoxicated, it produces what's called flaccid paralysis. And that means you can't contract your muscles because the botulism toxin blocks the release of acetylcholine. So it can't go across the synapse, and if it can't go across the synapse, then you can't contract your muscles. And a lot of people, there's usually around 150 cases of botulism in the United States each year. I think somewhere around 70-75% of those are infant botulism. And you need to, if you are a parent, a newborn baby, you need to be concerned with this. You do not want to feed your baby honey because honey contains the endospores of Clostridium botulinum. Can't say that enough. Do not feed your infant honey. And that's anything that contains honey. So you go out to a restaurant and you think, well, I'm just gonna mush this up and I'm gonna give it to my little baby, because they're like 6-7 months old, and you want to give them some squash, something like that, or some sweet potatoes and stuff like that. Make sure that they didn't use honey in any of their ingredients, because of the endospores that are in there. So usually when kids are less than 1, you don't want to give them honey, less than 1 year old, okay, because they can develop that infant botulism in there. The other majority of the cases, besides infants, it's going to be from people that like to can their own food. If you don't actually can it properly, then what can happen is you can get botulism from that. We'll talk about a little bit later why that happens when people can their food. But like I said, with kids, don't give them honey. Make sure that whatever they're eating does not have any honey in it at all. Even if it's cooked, remember Clostridium botulinum can produce endospores and it will be able to survive the actual cooking process. So if you don't know what's in that food item, don't give it to your baby. All right, so let's talk a little bit about proper food preparation. Some of the things that you need to be concerned with. So when you prepare your food, I don't know how many of you guys out there have those wooden cutting boards. You need to stay away from them. And the reason why is because they're so porous. Okay, so if you cut any kind of meat on them, then the microbes that are on that meat are going to be on that wooden cutting board, and they're going to be on there forever, basically. You're not going to be able to clean it. And you don't think it's just meat. I mean, if you have contaminated vegetables, the same thing's going to happen. Like, for example, most of the listeria outbreaks are going to be from contaminated fruits and vegetables. So if you've ever had lettuce and celery, and I think cilantro had a problem with that in the past. You've got all these different types of vegetables and stuff, and you cut them on there. If it has a pathogen on there, then you're not going to be able to get that pathogen off. And a lot of times people will get food poisoning and other diseases from salads because you don't cook your salad. So if that cutting board is contaminated, everything that you get off there is going to be contaminated. So stay away from those wood cutting boards. But also when you look at synthetic cutting boards, don't keep a cutting board forever. You shouldn't spend very much on your cutting boards. I know that some of them look nice, but even those synthetic cutting boards, once you start cutting into them and they get all those little cuts and stuff, that itself will allow microbes to grow inside of it. It's going to be hard to clean. I can't tell you how many times cook with some spices and stuff, and I've cut my marinated meat on the cutting boards, and then you try and clean it afterwards, and what happens is the little cracks and stuff are still red, or whatever colored spice you had on there is going to be that color. Okay, and it's really, really hard to get that stuff up. So once they start getting all those little cuts and stuff like that, you need to replace them with another one. So that's why I just buy cheap cutting boards. Yeah, you can get them for less than $10 in different places. You use them and you dispose of them. That way you don't spread disease. And you don't want to cross-contaminate, right? If you do have that wooden cutting board and you're one of those people that refuses to get rid of it because it's nice and pretty in your kitchen, make sure that you only cut your vegetables on there, or you only cut your meats on there, whichever one. If I were you, I would actually choose to cut my meats on there rather than my vegetables, because or actually I know, if there's a man in the house, he's not going to follow the rules all the time. I know, because I don't follow the rules all the time. And pretty much most of the guys I know don't follow the rules. So you say only cut the vegetables on that thing. What we're going to do, we're going to cut meat on there. And you're not going to know about it, because we're going to do it when you're not looking. And then people are going to get sick from that. So if you have to have that wood cutting board, make sure you know it's contaminated and cut stuff on there that you're going to cook. So you'll actually kill the bugs that are on that stuff. Right? So try not to cross-contaminate. When you barbecue in batches. And what I mean by that is, I know that when meat gets put on the grill and it starts cooking, it shrinks. Okay, don't start adding raw meat with the cooked meat on there at the same time. Just do the first batch, get it completely done. Okay, move on, take that one off, put it away. Then you move to the second batch. Okay, put the new raw stuff on, let it cook. Okay, completely remove it, and then move on to the third batch. But don't put raw meat on with cooked meats. Not a good thing. Make sure you wash your tongs, whatever you're using to actually move the meat around when you're barbecuing. You should wash your hands too if you're using your hands. Be as clean as possible. Don't cross-contaminate. And you should be watching this stuff all the time. Make sure you cook your meats to the appropriate temperature. And each one is different. What I love about HEB is on the little bags, it actually has what temperature is appropriate for which meat whenever you're cooking it. And one of the things you don't want to undercook is pork. Never eat undercooked pork, especially in this area of the United States, because a lot of our pork comes from Mexico, and Mexico has problems with worms. Okay, that's even in the restaurants. I know a lot of restaurants like to cook their stuff medium rare, stuff like that, especially the pork because it's nice and tender. But You can get worms from that stuff, and you don't want to get worms encysting in your legs or in your muscles. It'd be painful when they start encysting there, and then or they could actually go into your brain and cause all kinds of problems with that. So make sure you prepare your food properly. Then after you prepare your food properly, make sure that you store it properly. Major mistake that people make is they'll make some, let's just say, a big old pot of beans. I love to make beans. Right? And when I usually make beans, I usually start, for some reason, I don't know, I can't sleep at night. It's like, I think 11 or 12 o'clock right now. I don't have a clock on me. But it's pretty late when I'm doing these recordings, so I'm a night owl. So I'll start my beans at 11 o'clock, and like 3 o'clock, they're done in the morning. I have my fresh bowl of beans because they usually taste really good when you first get them off and they're first done. And then what I do is I leave the pot on the stove. Go to sleep, leave the pot on the stove. Why? Because it's way too hot to put in the refrigerator. And people don't know why they shouldn't put hot stuff in the refrigerator. A lot of times they say, well, it's because the beans that you just made are going to spoil. Like, no, that's not the reason. The reason why is because everything else in your refrigerator is going to spoil. So if you think about how long it's going to take for that pot of beans or that soup or whatever you made, that big old pot, to actually cool down, it's going to take a long time. And you look at that refrigerator, it's a small little box, right? You got a compressor that's regulating the temperature there, pumping cold air and getting rid of the hot air. Right? So there's only a small space. If you put a big old pot of beans in there, that's a huge source of heat inside of there. And that refrigerator is going to heat up, and you'll actually hear your compressor in your refrigerator kicking off, and it'll stay on for a long time because it's not going to be able to actually match the heat produced by that big old pot of beans or soup that's in there. And so what's going to happen is the temperature inside your refrigerator is going to be off. It's going to be higher for a certain amount of time. And if you have milk in there, eggs, different types of cold cuts and stuff like that, these microbes will grow at a fast rate because of the elevated temperature. The next time you open up your milk, it's not going to be very good, even if it's within the time that it is good. Even if you haven't even opened it, you just got it from the store. Put that pot of beans in there, and the next day it's probably not going to be very good because those microbes inside of that milk have been growing for a long period of time at the elevated temperature. Right, so you want to wait for the stuff to cool down. There's all kinds of things you can do to help it cool down faster. You can put it in smaller containers with large surface area to help it cool down. But you don't want to stick it in the refrigerator when it's really hot because that'll mess up everything else that's in your refrigerator. Right? Um, Let me see. Other things for proper food preparation. I think that's pretty much it for now. Oh, when you store your food, think about how long you store it in the refrigerator. I always ask my class, and I was like, how many people like sweet tea? When you make tea, put sugar in it. Think about that. That's nice broth for microbes. And so if you're leaving it in there forever, okay, what's going to eventually happen is you will notice that microbes are growing on there. You'll notice it because there'll be usually like a little oily sheen on the top. Okay, other stuff from the bottom. There's some stuff from the bottom when you pour it. That stuff comes up, and that's going to be the microbes that have settled to the bottom. There's going to be a difference in taste. It's going to have a strange aftertaste when those microbes start growing. So usually after day 3, what happens is those microbes have become high enough in concentration to affect that tea or whatever you've made. Okay, and so even though you put the stuff in the refrigerator, you know a lot of these things have a shelf life. Milk has a shelf life. So you open it up, it's only good for so many days. A lot of times people say 7 days, but it really depends and how contaminated that milk was. It's gone through pasteurization. Sometimes it's good for 7 days, sometimes it's not. It's going to be less than that. Okay, so what really sucks is usually the way you find out is you drink the bad milk and it's not good. Same thing with cold cuts. A lot of times cold cuts, whenever they start going bad, what happens is you'll notice a smell and you'll notice them getting slimier than they already are. So hopefully you notice that stuff before you actually take a bite of your sandwich. But those cold cuts have a lot of preservatives on them, which are antimicrobial agents. But still, these microbes are able to grow. And so a lot of times, like, let me go back to listeria. A lot of times when women are pregnant, the doctor will tell them to avoid cold cuts, avoid the hams and stuff like that. They'll say also unpasteurized cheeses and milks and stuff like that. Avoid that, and that's because of listeria. Even though those things have all kinds of preservatives inside them, the cold cuts, listeria is still able to grow on them. And as I said before, listeria can cause miscarriage, spontaneous abortion, that pregnant child. So you want to make sure and avoid that stuff. If your doctor hasn't told you that in the future when you get pregnant, do not eat those cold cuts and stay away from those unpasteurized cheeses. Our next physical requirement is going to be pH. Just like with optimal temperatures, these different microbes will like different pHs. And that's usually going to be based upon their normal environment. And so most bacteria like it between 6.5 and 7.5, right around neutral. Molds like it a little bit more acidic. And the reason why they like it a little bit more acidic is because they have less competition. If you think about the bacteria, they divide once every 15 to 20 minutes, so they're very, very fast growers. The molds and yeast, sometimes they take an hour to divide, sometimes longer than that, because they're eukaryotic organisms, so they just take a little bit longer. So if they like it in a range where most bacteria don't like it, then they'll have all that food to themselves. They will have minimal competition. So a lot of times you think about molds and yeast, you usually commonly find them growing on your fruits, right? Because there's citric acid there. It's too acidic for the bacteria, so they'll contaminate your oranges, those bastards, and grapes and things like that. But you won't find a lot of bacteria there because it's way too acidic for them. It's really the different areas of your body. The cytoplasm of the cells is around 7.2. Blood plasma is right around 7.4. So there are a lot of different areas in the body where it's right around neutral. So we will find the most pathogens are neutrophils, like it right around neutral. If they like it acidic, then they're acidophiles. If they like it basic, they're alkaliphiles. So these are three terms that are nice, should be nice and easy to remember. Acidophile, neutrophile, and alkaliphiles. Helicobacter pylori is a pathogen that causes peptic ulcers. And you can see a peptic ulcer right here in the stomach. But it doesn't, or it actually is one of those weaker pathogens that actually needs some help. Say it doesn't do it on its own. Way back when, we used to think that stress caused ulcers, or coffee caused ulcers. But now we know that it's actually a combination of things that help to weaken the immune system, and that weakened immune system will allow Helicobacter pylori to actually cause damage to us. So you have the stress, which does a number on your immune system. Coffee, with all the, you're probably stressed and working strange hours and stuff like that if you've got coffee inside of you, and so that weakens your body and allows this particular bug to do damage, to grow enough to do damage to your body. In order to get these microbes to grow, we also have to provide them with the appropriate osmotic pressure, salt concentration. Some of these microbes don't like very much salt. They're non-halophiles. Some of them like salt, and so they're halophiles. The salt-loving microbes are called halophiles. And there's two different types of halophiles. Okay, there are the obligate halophiles, and then there are facultative halophiles. That term obligate means requires. Okay, they have to have it. Some of them actually have to have a huge amount of salt, 20-30% salt. Yeah, they like that because most microbes don't like salt. If there's too much salt, what usually happens is plasmolysis. Right? And so, if you remember, we talked in chapter 5 about osmosis. Okay, if there's a high concentration of salt on the outside, that means higher than what's on the inside, then what happens is water flows out and the cell shrivels up. The end results of plasmolysis are going to be death or hibernation. Okay, one or the other. And either way, the microbes aren't growing. And so most microbes don't like that. And so if these bugs like 20-30% salt, all the nutrients in that area are theirs. Nobody else is going to like it. They don't have any competition there. Okay, so obligate halophiles require that high salt concentration. Facultative halophiles tolerate high and low concentrations of salt. Okay, so they just can live in different areas. So an example of a facultative halophile would be Staphylococcus aureus. Staphylococcus aureus can grow in low and high salt concentrations. And if you think about what happens on the surface of the skin, okay, it's apparent why it needs to be a facultative halophile. So in the morning, most people usually get up and take a shower. Right? As you take a shower, what you're doing is you're washing the salt off of your skin. So you have low salt concentration. As you're going throughout the day, what happens is you are practicing homeostasis. In order to practice homeostasis and keep your body temperature regulated, you're going to sweat, and that sweat's going to release salt. So all throughout the day, what's happening is the salt concentration in your skin is going up. And so Staphylococcus aureus is found on the skin and nasal mucosa, and it needs to be able to tolerate both high and low concentrations of salt because that's what happens on the surface of the skin. Every time you wash your hands, what happens? Salt concentration goes down. Every time you take a shower or a bath, salt concentration goes down. All the rest of the time, salt concentration is going up. Now, what really sucks about Staphylococcus aureus is that it's found in many places, and because it's a facultative halophile, it's growing in areas where we don't think a lot of bad bugs can grow. Like a lot of people, it used to be. Now we're becoming more aware, or people are becoming more aware. A lot of people think, well, go to the beach and it's so salty in that water that a lot of microbes can't grow. And that's a bunch of crap. We know that nowadays because that's at the beach, inside the water. That's a common place to actually get MRSA, methicillin-resistant Staphylococcus aureus, because MRSA is a strain of Staphylococcus aureus, which is a facultative halophile. It can grow inside of salt water. It can be on the beach where all the sand is, and that's all that's in the sand. But it's also in the water, so you have to be very careful when you get in that water if you have any cuts and stuff, because you can contract a staph infection when in the water, and hopefully it's not MRSA. But there are also other bugs out there that are facultative halophiles. As I said before, these bugs can be really bad. Now, if we look at these two things, and some other things that are in your house. Okay. A long time ago, when I used to live in my mom's house, she used to put the peanut butter in the refrigerator, and I hated that. Right? Because like you cannot spread peanut butter once it's in the refrigerator. We're talking about the normal peanut butter. I'm not talking about the freaky stuff that's all oily and stuff. So I'm talking about the normal, non-healthy version, right? So you get the Jif, you put it in the refrigerator. Can't spread it on the bread, but you don't have to put your peanut butter in the refrigerator. A lot of people do it because they think, well, once you open it up, microbes are going to get in there and you're going to have some problems with that. The reason why you don't have to put it in the refrigerator is because it has a high solute concentration. Okay, those bacteria, if they do land in there, it's going to be there are going to be too many solutes inside of there, and so they'll undergo plasmolysis. So when we talk about halophiles, okay, a lot of times our common term for halophiles is salt-loving microbe, but they actually love high solute concentrations, not just salts. There are other solute molecules that are there. Okay, so peanut butter contains a bunch of solute molecules. It contains a bunch of salt in there too, but it's too high in concentration for most bugs to like it, and that's why it doesn't go bad very quickly if you leave it out. Okay? And you can leave it out for a long period of time and it won't go bad. Okay? It's the same thing with your jams, your jellies. They have a high solute concentration. I usually like my jelly cold, so I keep it in the refrigerator. One thing you do have to be concerned with the jams and the jellies are fungi. So if fungi get on there, then they will be able to grow like crazy inside of there. With honey, it's the same situation. You don't have to keep it in the refrigerator because its solute concentration is really high. It's very viscous, which shows that it has a high solute concentration. Once again, I told you, you do have to worry about a certain pathogen with that when you have little kids, because those little babies are immunocompromised. If they're less than 1 year old, you don't want to feed them the honey because it has Clostridium botulinum in it. And it's not the vegetative form, it's actually the endospores of Clostridium botulinum that are in the honey. So the vegetative form of Clostridium botulinum cannot survive in honey because it's too high in solute concentration, but the endospores can. And those babies can't break those endospores down and destroy them like we can. A lot of people think, well, how come babies have a problem with this and we don't? It's because when you look at the baby's digestive system and its immune system in general, it's not as developed. So think about this. You feed a baby carrots. What color is its crap? Crap is orange, right? You feed it peas, the crap is green. Right? It has these different colors, and the baby's gonna have different colored crap. With us, you would actually have to eat a boatload of carrots to actually get your crap to turn orange. Most of the time, it's not, right? It's gonna be brown. It's gonna be the same brown color. That's because your digestive system breaks all that stuff down. It's the same thing with peas and stuff like that. You will eat certain foods that can make your crap certain colors. Like I've noticed past year started eating Fruit Loops again, and when I eat Fruit Loops, my crap is green. It's like really green. Freaked me out the first time. I was like, "What the heck is going on down there?" But that's what happens. So it's the dyes that are in there. It turns green. So it's pretty interesting. You should actually try it out. Let me know what's going on with you. If it's the same thing happens with you. So you look at the, like I said, the honey, way too high solute molecule concentrations. You don't have to keep it in the refrigerator. You can just keep it in the cabinet. And eventually, yeah, things will break down and go bad. I just threw out some honey today that was like crystallized and stuff like that. So it wasn't very good. I think it went bad in 2013. Don't judge me. Like I said before, Staphylococcus aureus isn't the only facultative halophile that causes problems. This Vibrio vulnificus is a microbe that's becoming more and more famous off the coast, the Gulf Coast. It is there are certain strains that are flesh-eating. So this guy right here this story, he went out wade fishing like he normally did. He was in Florida, and he had some cuts on his legs, and he ended up getting very, very sick and dying because of the infection that he encountered. And it was this flesh-eating bug that's there. Just this last year, one of my I don't even know what he is. He's like a relative's brother. Okay, he kind of got in the family through marriage. He has a charter fishing operation that operates out of Texas City, and he got a cut on his leg, just a little bitty cut on his leg. Didn't think nothing of it. The next that evening and the next day, he started feeling bad, and he was like, "Oh, I have some customers, so I have to go out." So he goes out, and a couple hours into the trip, he starts vomiting, and he's vomiting because he's in so much pain. And his leg was dizzy, and he had a huge headache, and just really bad. So his brother goes down there and takes him to the Texas City Hospital, and they look at his leg, and his leg is starting to swell up and stuff like that. And so he goes to the emergency room, and he's telling them, he's like, "Hey, I need you to test me for flesh-eating microbes because this is where I work. I work in the fishing industry, and this is what I think I have." And they're like, "No, no, no, you don't need to worry about that." So they just gave him some antibiotics, and they sent him on his way. If somebody doesn't treat you the way you want to be treated, you need to make sure that you keep on going to the hospital. Go to a different hospital. And that's exactly what he did. He went down to Bentop, and he told him, "Hey, I'm not feeling very good." He had a discussion with his brother, and his brother was like agreeing with him totally. He's just like, "Look, we're just going to end up taking you back to the emergency room because you're in bad shape still, so we need to deal with this." So he went to Bentop. He tells him, "Hey, I'm working in the fishing industry. I need you to test me for these flesh-eating microbes." And they said, "Okay." So they did it. And it turned out that he actually had flesh-eating microbes that were in his wound, and they were starting to spread. And so they loaded him up with antibiotics, and it was almost a month later he got out of the hospital. And I don't even know if he's returned to work yet, but this happened over the summer, I think either June or May. But he was out of work for a long time, and he's actually lucky. The doctors were telling him he's lucky that he went back to the hospital because he could have lost his life. And I saw some of the pictures, and I wish I could put them up here, but um, I don't actually have access to them from my cousin's phone, and it was his leg had doubled in size, and there was a lot of pus, and it just looked really, really bad. Like I said before, if you don't think you're receiving the right treatment, make sure that you go to a different hospital and complain enough where you do receive the right treatment. Don't be afraid to check yourself out of one place and go to another one. It'll treat you right. It's better than dying. Right, now we're going to go into the chemical requirements for growth. Carbon. Carbon is an important molecule. We are actually carbon-based life forms. So if you think about the macromolecules that we talked about in chapter 2, proteins, lipids, carbs, nucleic acids, okay, every single one of those contains carbon, and there's actually a lot of carbon. So organisms get their carbon from one of two ways. They're either autotrophs, which are self-feeders, or heterotrophs, which are eaters of others. Okay? And sometimes what these bugs will do is when they're eating other things, they will actually live on dead or dying organisms, and those are a group of heterotrophs called saprobes, and they're very, very important. A lot of times the fungi are saprobes, and what they're doing is they are actually recycling nutrients. So when an organism dies, it's important to get those nutrients back into the food chain, right? Because somebody's got to break that stuff down so that other organisms can use it. Because we only have a finite amount of nutrients on this planet, so it's really, really important that we break down those other organisms that have already passed. And so you'll have other organisms eating them and using them for nutrients. All right, so if we look at this structure on the right over here, this is the DNA molecule. All those gray spheres in there are the carbons. There's tons and tons of carbons in DNA. The blue spheres that are in the middle, those are the nitrogens from the nitrogenous bases. Okay, nitrogen is also found in proteins. And so with these microbes, whenever they need nitrogen, one of the things they can do is break down proteins, okay, or they can break down nucleic acids. Another thing, if they have the enzymes to do so, is they can carry out nitrogen fixation. So if you look down here, okay, you can see these little balls that are on this root system. Those are called root nodules. And there are certain groups of microbes, which we'll talk about in chapter 11, which can associate with plants. They're called rhizobium. Okay, and they carry out a process called nitrogen fixation. Now, nitrogen is actually very abundant on this planet, but it's actually into nitrogen, molecular nitrogen, which is gas. And we don't have the enzymes to use that version of nitrogen. Our nitrogen that we can use actually contains some hydrogens or some oxygens on it. It just depends on which enzyme you're talking about. But we need some stuff to be added on to this nitrogen in order to make use of it, put it in our proteins, our nucleic acids, and stuff like that. There are some organisms that can directly pull this stuff out of the air and turn it into a usable form. And that's what's happening right here in this root nodule system. The rhizobium are in there. They are pulling out the nitrogen. And basically what happens is this is a mutualistic relationship, which means that both organisms benefit this relationship. So one of the things that's limiting the environment is usable nitrogen, also usable phosphorus. But here's this is the nitrogen part here. So even though there's plenty of nitrogen in the air. And so these plants need nitrogen. If you look at most of the fertilizers out there, they're loaded with nitrogen because you know that they need some and it's lacking in the environment. But these groups of organisms that can interact with the rhizobium, we'll talk about them a little bit, like I said in chapter 11, they're called the legumes. They can actually form this relationship with rhizobium. And so what happens is the rhizobium are in the soil. These legumes, they produce their roots and they make these little root nodules where the rhizobium are. Because they know that the rhizobium are going to give them nitrogen, and in exchange for that, the plants are going to give the rhizobium a place to stay. They're going to make different types of amino acids and stuff for them, and basically provide them with nutrients. And so the rhizobium basically say, I'm going to give you all the nitrogen you need, and then some. You give me a place to stay, and that's what happens. Okay, that nitrogen fixation can only take place in those little root nodules in there. And so the plant provides them with that environment where they can carry out nitrogen fixation. Right, phosphorus is going to be in phospholipids. Phosphorus is also going to be the orange spheres in that DNA molecule. So in order to get phosphorus, they can break down the phospholipids or they can break down nucleic acids. We're also going to have to provide these bugs with what are called trace elements. Okay, a lot of times these trace elements are cofactors. Remember, cofactors, they help enzymes do their job. So things like magnesium, zinc, calcium, things like that, they're going to be needed. And the reason why we call them trace elements is because we only need them in small amounts. Now, a lot of people don't realize this, but when we're breathing the air, we're actually breathing in a lot of toxins. Not just toxins that we make, but toxins that are naturally there. And so one of the things that we actually need in order to survive is oxygen. But the bad thing about oxygen is it actually combines with things, it breaks down, and it forms, or there are actually several different forms of oxygen, and many of those different forms are actually toxic. So anytime you have molecular oxygen around, you'll have different other forms of oxygen around which are toxic. Okay, and some of these things are here, like singlet oxygen, superoxide free radicals, peroxide anion, hydroxide radical. These are all different toxic forms of oxygen. And what these things do, these toxic forms of oxygens are electron stealers. So they steal electrons away from molecules, they break covalent bonds. So any organism that lives in an oxygen-rich environment is going to have to be able to neutralize these toxins in order to be able to survive. And so since we live in an oxygen-rich environment, we require oxygen, we have the enzymes to neutralize these different toxic forms. Because if we didn't, then the toxins would kill us by destroying our molecules. And so we have things like superoxide dismutase, which takes that superoxide and it breaks it down into hydrogen peroxide and molecular oxygen, which is a normal version that we actually need. But the bad thing about hydrogen peroxide, it can break down into the peroxide anion, so this H2O2 is going to need to be dealt with, and we have the enzyme catalase, which breaks hydrogen peroxide down into water plus the non-harmful version of oxygen. And you can see the action of catalase. If you've ever put hydrogen peroxide on your wound, you've seen the bubbles because of the production of that oxygen that's there. Some organisms actually have peroxidase, which breaks down hydrogen peroxide into just water. Okay, but we actually have catalase. And so if we look at these microbes, the next slide you'll see microbes growing in different areas of the tubes. And I've tried to, in this slide, explain why they're growing in different areas of the tube. Okay, so the molecule, these different microbes will have to make ATP, and there's two different ways to make ATP. So right here is my tube. Okay, here's the broth, and microbes will be growing in different areas depending upon how they make their ATP. That's one of the reasons why they're going to be growing in different areas. Now, aerobic respiration is only able to to be performed whenever oxygen is around. So if we look at the concentration of oxygen, it's going to be high, and as you go to the bottom, you get down to no oxygen. So only when there's oxygen around, that's when where aerobic respiration is going to occur. So this area right here. Anaerobic respiration doesn't depend upon oxygen at all. And so it doesn't care if it's there or not. So anaerobic respiration can occur throughout the entire tube, but aerobic respiration can only occur in areas where the concentration of oxygen is pretty high. Okay? As it goes down, then so does the ability to perform aerobic respiration. Now, remember what I said before. Anytime you have oxygen around, you also have toxic O2 around. And so when there's a high concentration of oxygen, there's a high concentration of toxic O2. And you can see that as you go all the way down to the bottom, there's a concentration gradient. So as you get lower and lower in the tube, you go from high concentration of toxic O2 down to nothing. Now, these organisms that we're going to be talking about, they either have the ability to neutralize the toxins, or they don't have the ability to neutralize the toxins. Okay? And so they'll be growing in different areas depending upon what they have. In order to grow up here in this area where there are toxic O2 molecules, they have to have the enzymes to neutralize the toxic O2, right? Because they're going to have to deal with the toxin. If they don't neutralize the toxins, they die. Okay? If they don't have the enzymes that are necessary to neutralize the toxins, then they have to live in an area where the toxins aren't present. And that means only at the bottom of the tube. Remember, there's a concentration gradient. Toxins are up here at the top. They're not there at the bottom. So you put these two together, you see why these organisms grow in these different areas. Okay, so when it comes to oxygen requirements, we have a bunch of terms. Remember that term obligate. Obligate means requires. So an obligate aerobe, like Pseudomonas, it's going to be growing up here at the top. The only way that it can get its ATP is through aerobic respiration. So no oxygen down here means no growth. No ATP means they're going to die. So they have to grow up at the top. And remember, up at the top, okay, not only is there oxygen, but there's also toxic O2. And so these obligate aerobes have to have the enzymes to neutralize the toxic O2 to be able to stay alive up there. Okay, the other guys, all the way on the other side of the spectrum, the obligate anaerobes, they are only growing down here. Okay. And you can see a difference between the obligate aerobes, which are over here. There's tons and tons of cells here in the top of the tube. They're undergoing aerobic respiration, which means they're going to have 36 ATP per glucose molecule. These obligate anaerobes are undergoing anaerobic respiration, which means they only get maybe 2 ATP, depending upon what type of anaerobic respiration they're undergoing. But you can see there's going to be fewer cells because remember, anaerobic respiration is less efficient. Now, they are growing down here because they do not have the enzymes to neutralize the toxic O2. And the toxic O2 is going to be all in this area. So it's way too toxic up there for them to grow, so that's the reason why they are only growing at the bottom, because they don't have the neutralizing enzymes. Okay, let's go over here to facultative anaerobes. Facultative anaerobes, you can see, are growing throughout the tube. What you should be able to see is that over here, up at the top, there are more cells. Because over here at the top, they are undergoing aerobic respiration. Fewer cells down here because they're undergoing anaerobic respiration down here. Remember, the toxins are going to be found in this region right here. And if in the tube, if there are any cells growing in that area, that means that they have to have the enzymes to neutralize the toxic O2. So since they're growing in that area, they do have the enzymes to neutralize the toxic O2. But these facultative anaerobes, as they said, they switch what type of respiration they're undergoing. Up here, it's aerobic. Down here, it's anaerobic respiration. That's why you have fewer cells down here. It's less efficient. More cells up here. Now, we're going over here to the aerotolerance. With the aerotolerance, you can see that they are growing equally throughout. They are only undergoing anaerobic respiration, and that's why it's equal concentration. They don't care about oxygen in terms of making their ATP. Okay, and anaerobic respiration can occur all throughout the tube, and that's why they're found all throughout the tube. But remember, the toxins are going to be in this section. So you see them growing in this section right here. They have to have the enzymes to neutralize the toxic O2, and they do. Okay, that's why they're able to grow up there at the top. Okay. But the difference, a lot of people are confused between facultative anaerobes and aerotolerant anaerobes. The facultative anaerobes switch. Up at the top, they're undergoing aerobic respiration. At the bottom, they're undergoing anaerobic respiration. Aerotolerant anaerobes are undergoing anaerobic respiration all throughout. They do not switch. Both of them, both the facultative and the aerotolerants, have the neutralizing enzymes. Now our last group, it's over here. So you can see the microaerophiles. The microaerophiles grow within a certain range in this tube right here. And the reason why is because they make their ATP through aerobic respiration. Since there's no oxygen down here, they aren't able to make their ATP down here. So if any of those cells happen to float down there, they won't be able to make their ATP and they'll die. Okay? Now, they're not growing up here because toxico-2 is in this area, and it's too toxic for them in this area right here. Concentration of those toxins are too high. They only produce a small amount of neutralizing enzymes. They only produce enough to deal with the levels of toxico-2 in this area. Because remember, we talked about how, in the last slide, we talked about how there's a concentration gradient. It's high up here, none at the bottom. Just go down. So if they only have a certain amount of enzymes, they'll only be able to tolerate a small amount of toxin, which is that amount right there. Okay, so if any of them float up, the toxico-2 levels that increase are going to kill them. If any of them float down, the lack of ATP is going to kill them because they can't undergo anaerobic respiration. So those are the microaerophiles. All right, so we have all these different requirements that have to be met in order to grow microbes. Remember I said we can grow less than 10% of the microbes out there. A lot of these microbes have special dietary needs, but it's important that for us to be able to grow these microbes. Now, when I give you media, that media has gone through a lot of stuff before you actually get it. Okay, the lab coordinator takes a powder that she buys from a company, mixes it with water, and then she puts it through a sterilizer. It's important that it be sterile because if it's not sterile, then you'll have a bunch of bugs growing on there already. And who knows what the heck is going to be on there, and it could be pathogens that make you very, very sick. You want to be able to only put what you want on that plate. So that's why it's important to be sterile. Once you inoculate the plate, you have to provide these microbes with the appropriate minimal conditions for them to grow. All you have to do is meet their minimal conditions. Proper pH, moisture, oxygen concentration, different chemicals that are on there, trace elements, all that kind of stuff. You're going to meet for these bugs and then they'll be able to grow. Remember what I said, we can grow less than 10% of the microbes out there, so it's actually hard to get these bugs to grow. Okay. These bugs that have special dietary needs are said to be fastidious microbes. Okay, and you think about it, we can grow less than 10% of the microbes out there, which means that over 90% of the bugs out there are fastidious. One of those that's easy to grow is E. coli. As I said before, we've got different chemicals that we put into water, and we can make just a general purpose media for them. So if we look here, glucose. We talked about how glucose is an energy source in chapter 5 when we went through cellular respiration. It's a carbon source. Okay? And so we put a certain amount in there so that these bugs can make ATP. Ammonium phosphate, we need a source of nitrogen for our nucleic acids and also for amino acids. And we need phosphorus for our nucleic acids, phosphate groups, and for our phospholipids. Sodium chloride, that's for osmotic potential, to make sure that they have the appropriate amount of salt that they need. Magnesium is an important cofactor, and sulfur is found in certain amino acids. Potassium is another cofactor, and phosphorus, we already talked about what that's there for. So we have all these different things, these different nutrients in here. With these potassium phosphate, ammonium phosphate, these are also buffers that help us to obtain the appropriate pH concentration inside of there. And then we add water. Remember, the only thing that limits life is the availability of water. So this is a nice broth we could make that meets E. coli's minimal growth conditions. So this is all that it needs in order to be able to grow. E. coli can make everything else for itself. But if it couldn't make everything or something else for itself, then we'd have to provide it in our media. And sometimes we have to provide some strange stuff like liver extract. You'll see some bugs growing on blood as well a little bit later in the semester. Sometimes yeast extract, things like that. So sometimes they need some extra stuff. If we wanted to make our media semi-solid, we could add agar to it. Now, agar is great because most microbes don't break it down. I've seen some in the past that were able to break it down. It's kind of weird when they do that. But most microbes, they'll just grow on the surface. So it's kind of like Jello. and we can actually add some stuff to the agar to make different types of media. So what's great about agar is it liquefies at 100 degrees Celsius, but doesn't solidify back down until it gets to around 42 degrees Celsius. What's great about that is that some of the items that we need to add to our media are sometimes heat labile, which means that if you put them in and it gets too hot, that they get destroyed. Great example would be certain antibiotics like penicillin. Okay? And other versions of antibiotics. A lot of them are actually heat labile, so they'll break down when it gets too hot. So let's just say that we wanted to be able to only on this plate right here, we wanted to only be able to grow those microbes that were resistant to penicillin. How would we do that? Well, we'd make our nutrient agar. We'd add all the powders, let's just say all that stuff from the last slide, into a flask. We have the appropriate amount of water, then we have the appropriate amount of agar, usually depending on how much we're making, about 6 to 12 grams of agar. We mix it all up, put it in the autoclave. Okay, the autoclave gets up to 121 degrees Celsius, usually between 15 to 30 minutes. Okay, 10 to 15 pounds per square inch pressure is going to be there. And so what comes out is sterile. We're going to let it cool down, and once it gets to around 50 degrees, we'll add some penicillin in there, mix it up, and then we'll pour it into these petri dishes. It provides us with a lot of space. Okay, if we let it solidify, the next thing we can do is we can inoculate this with a sample. Let's just say we got a patient, we get a sample from a patient, and we just streak that sample onto this plate. The only thing that's going to be growing on that plate, if penicillin is in that plate, is going to be the penicillin-resistant bugs. We'll do it a little bit differently in class. In class, we'll have general purpose media, and we'll put little antibiotic coated discs on the plate. Okay, but a lot of times in biotechnology, we will make tons and tons of plates that have penicillin inside of them, or ampicillin, or other antibiotics, chloramphenicol, stuff like that. Other things we can do with our media is we can put it in tubes. So in the middle here, we have our broth, which is nice and liquidy. Here we have what's called a deep. So you pour the liquid in there, it's got agar in it, and it solidifies. It's nice flat. The broth doesn't have any agar in it, so it's nice and liquidy. And we usually use broth to actually grow our microbes very quickly. So a lot of times it's overnight. With our deeps, a lot of times we will stab the microbes down there, and we'll check for things like gas production. You'll be using slants a little bit later. So the slants have two parts. This bottom part is called the butt, and then this top part, which is right here, is called the slant. And you'll be, a little bit later, you'll be stabbing microbes all the way down here to the bottom, and then once you get up here, you're going to fishtail and inoculate the slant as well. And we'll have different types of media with different nutrients in the different areas of these slants. Right, so with general purpose media, we use that to grow a bunch of different types of microbes. If we don't know what's in our sample, we want to use general purpose because that'll allow us to grow a bunch of different types. If we want to get rid of certain types of microbes, then we use selective media. Okay, selective media usually kills one or a few types and allows, sometimes it only allows one type of microbe to grow, sometimes it allows multiple groups of microbes to grow. McConkey's agar actually kills off one major group of microbes and selects for the other. So it kills gram positives and it selects for gram negatives. So all the microbes that are growing on here are going to be gram negative. And the way it does that is it contains crystal violet and bile salts. And if you think about the difference between the cells, gram positives have a thick cell wall, the gram negatives have a thin cell wall. Those crystal violet and molecules and bile salts, what they do is they get tangled up in the thick cell wall of the gram positives. And that means no nutrients can come in, waste can't get out, and so those cells will die. The gram negatives, they don't have that problem because they only have a thin cell wall, so they're able to grow. So that's McConkey's is selective. But McConkey's is also differential. So if you look at this plate right here, you can see that the individual colonies are kind of maroon in color. If they're maroon in color, what that means is this particular bug can utilize lactose. Sorry, there's a noise here for some reason. But this bug can utilize lactose. If not, then they're a nice creamy color. Okay, so what you know about the microbe on this plate is that it can utilize lactose and it's a gram-negative organism. And besides being selective, some types of media are differential. And what that means is they'll allow a couple of different types of microbes to grow. And when they grow, you can see things that they can or cannot do. So this one over here on the left is MSA, mannitol salt agar. Okay, it contains usually around 7.5% salt. So the bugs that are going to grow on there are going to be halophiles. So you look the bottom left here, and this guy right here is a halophile. This one did not grow, so it's a non-halophile that was inoculated right there. Okay. Not only is it selective, but it's also differential, because with these two bugs that grew, okay, the normal plate color is kind of pinkish fuchsia, which looks like this on the right. Okay. If the organism could utilize mannitol, it's going to produce acid. Okay, and the reason why the plate was kind of fuchsia color to begin with is because it has a pH indicator inside of it. So if it produces acid, the plate will go from that pinkish fuchsia color to an orangish yellow color, okay, like shown on the left here. Okay, so this bug that's down here on the left, you know two things about it. First, you know since it grew, it's a halophile. Second, since there was a color change, you know that it can utilize mannitol. The one on the right here, this one, you also know two things about it. One, it's a halophile because it grew on this plate. And the second thing you know about it is it cannot utilize mannitol because the media was the same color to begin with, so it didn't produce any acid that was there. So that's differential media. You see two microbes that grew, and you have different results based upon what types of metabolism they carry out. Over here on the right, you have blood agar. Blood agar is used to check for hemolysis. Hemolysis is the lysis of red blood cells. And some of these bugs will tend to lyse our red blood cells to steal the iron that's on the inside. So if you remember in chapter 5, we talked about how transferins hide the iron from these microbes. They hide the iron because the iron is an important cofactor. Some of them, their metabolism will be slowed down, which slows down their rate of growth or their rate of toxin production. Okay, but some of these microbes have the ability to get some iron back by breaking down our red blood cells. And so that really, really sucks. We've got three types of hemolysis. We have beta hemolysis, which is over here at the top. That's complete lysis of our blood cells. So you can actually see it clearing around where this microbe is growing because the media is red because of all the blood cells that are in there. This is blood agar. It's nutrient agar supplemented with 5% sheep's blood. Okay, so these types that can carry out beta hemolysis do a really good job at destroying all the blood cells. Alpha is over here. These are the Greek symbols. So this symbol for alpha, that's partial lysis of the red blood cells. Okay. And so it can break down some of them. Doesn't do as good a job as beta, but it can still break down some of them. And then over here on the right, that's gamma. Gamma cannot break down the red blood cells, so it can't carry out hemolysis. As I said, our defense against some of these pathogens is to hide the iron from them. They, in turn, some of them can actually carry out hemolysis, which lyses the red blood cells, and they'll get the iron back from our hemoglobin that's there and defeat our defense. Now, 2 out of the 3 can actually defeat our transferrin defense, and that's going to be the ones that carry out beta hemolysis and the ones that carry out alpha. Right? Iron is a trace element, and the reason why alpha can defeat our defense is because it carries out hemolysis. Not very well, but it still carries out hemolysis. And so it can get some iron back. It only needs a very small amount to actually be able to defeat that defense, since iron's a trace element. And so we'll be seeing these different types of media a little bit later in the semester. But remember, it's very, very important to be able to grow these different microbes on the media. We can grow less than 10% of the microbes out there. And once we grow them, we have to identify them. So we're going to have to inoculate different types of selective and differential media to be able to get that metabolic fingerprint that I was talking about in chapter 5. So we can identify them. But along with doing that, we'll also have to do sensitivity tests. We'll make different types of media and add things like ampicillin, tetracycline, all these different amoxicillin to them to see what these microbes are sensitive or resistant to. We want to get a complete profile. We don't just want to identify them, we want to figure out how to kill them. And so that's what the lab people are going to be doing once you give them that sample.